Hey, physiology students, welcome to Monday's lecture on Chapter 2, Part 2, Cell Transport, or how a cell transports material into the cell or out of the cell. Okay, so what we're going to do is we're going to start with the PowerPoint. I'm going to refer to our wiki textbook as well, and also our online resources. So I'm going to share the screen. Hope you all are doing well. Okay, we're getting close to in-person teaching, so we've got next week of online classes, and then the week after, we're going to actually meet each other. Okay, so let's start with the PowerPoint, and I'll get to the wiki textbook in a second. But here is Chapter 2, Part 2, Cells and the Environment. Okay, so we're going to talk briefly about cell membrane permeability. A lot of this is going to be repeat of your intro biology courses. If you took it here at Francis Marion, it's going to be Biology 105, cells, genetics, and passive transport, active transport, things like that. Okay, and then later on Fridays or Wednesday's lecture, we will talk about membrane potential and action potentials as a prelude to next week's lecture on neurotransmitters and how they're used to stimulate or inhibit cells. Okay, but let's just talk about cell membranes. Okay, so if you have a permeable membrane, a freely permeable membrane, what you'll have is substances will move across the membrane freely without channels or receptors or anything like that. It's kind of like there's no bouncer at the bar. The door's just wide open and you can go and come as you please, okay? So primarily gases work this way. Cell membranes are highly permeable to gases like oxygen. That makes sense. Our cells need oxygen. We've talked about that in metabolism. We need to get rid of CO2 waste that's produced from cell metabolism. And there's other gases like nitric oxide. But that is going to go across your cell membranes without any restrictions. Most of our cells, however, work by a selectively permeable membrane. So we're talking about charged ions like sodium ions, calcium ions, potassium ions, chloride ions. They all have a charge associated with them. Large particles are going to have difficulty moving across a membrane. It's not impossible, but they're going to be more restricted. Even glucose, which all of our cells need, right? There's actually a bouncer at the cell membrane. There's a receptor that the glucose has to bind to and has to open up to allow that glucose in. So if you look at the GIF here below, and I will get my laser pointer, what we've got is we've got water molecules, the smaller molecules that can go across the membrane, but the larger molecules, they could be glucose, they could be anything. Notice they're not getting across easily. All right, so that's why I have the picture of the doggy trying to get through the kitten-only door. A kitten small is going to get through. A doggy, not so easy. So you can think of that as a semi-permeable membrane. All right, so let's move on. Now, there's going to be two types of cell transport. This is going to be review of intro bio. There's going to be movement of substances across a membrane that don't require energy, and that's called passive transport, all right? And that's what makes it passive. And then there's going to be active transport, where you're moving substances that are either large or are charged ions or in some other way are restricted in their entry, and it's going to take energy or ATP. So passive transport, I have the image of the guy riding downhill because you're coasting. You're going with the concentration gradient. You are going from one side of the membrane where solutes are in high concentration to across the membrane where those solutes are in low concentration. Now, those solutes could be sodium, potassium, glucose, almost anything, okay? And since it's going from high to low with its concentration gradient, it passes passively through the cell membrane. Okay, and we're going to talk about 5 forms of passive transport shortly. Now, the opposite of this is if you move substances from where they're in low concentration across the membrane to where they're in high concentration, that's going uphill. That's going against the concentration gradient. Think of it like swimming against the tide, or think of it like salmon swimming upstream where they're moving against the force of water. That takes a lot of energy. All right, so active transport is going from high concentration, sorry, low concentration to the side of the membrane where those are in high concentration. And so it's going to take energy often to pump. That's what this thing is supposed to be representing, a fueled or energy provided pump that will pump stuff from low concentration to high. Okay, so you're going uphill. It's going to take ATP. All right, so let's look at the forms of passive transport. Simple diffusion. You're going from high concentration across the membrane to where it's in low concentration. So no energy, no carriers, no receptors, nothing. All right. So gases certainly move this way. Small particles can move this way. Even though glucose is, well, we'll talk about that in a second. But basically remember high to low, going with the concentration gradient. No energy required. So let's look at another type of diffusion. Simple diffusion with channels is, well, think of it as a door that basically has to open up to allow you through, and the wall would be the membrane. Okay, so charged particles tend to move by simple diffusion. So sodium, notice the positive charge, chloride, the negative charge, potassium, the positive charge. Now, cells are pretty particular about charged ions coming across, so they can diffuse, but they have a channel. Now, in the next chapter, which is chapter 4, we're going to talk about how neurotransmitters bind to these channels and allow them to open up so that charged ions can get through. But I'm a little ahead of myself, but remember that simple diffusion with channels, okay? Moving from high to low, but through a channel or a door that has to open up to allow them through. The next form of passive transport. Facilitated diffusion with carriers. Glucose and small proteins, amino acids, work this way. All right, so what happens is they bind to a carrier, and then the carrier, usually like a rotating door, will turn around and spit them into the cell. All right, so carriers. It's facilitated diffusion. It's not an open door, but more like a rotating door. Okay, so remember, glucose and small proteins work this way. Okay, osmosis. We're going to have an entire lab based on osmosis and diffusion of particles across the membrane. So when I think of osmosis, I look at the O in osmosis and I think H2O, water. Osmosis is the movement of water across a membrane from where the water is in high concentration to where it's in low concentration. Well, here's another way of thinking about it. So if you've got water on two sides of a membrane, and you've got some solutes on two sides of a membrane. Whatever side of that membrane has more solutes dissolved in the fluid, that could be salt, it could be sugar, it could be just about anything. The water is going to want to cross the membrane where the side that has the solutes in greatest concentration in order to dilute those solutes, okay? Water wants to dilute things, and so it's going to move across the membrane to where there's more solutes to dilute, all right? So on your own time, go ahead and look at this GIF, and it's showing you the water being the small molecules and the sugar being the large molecules in green. And you obviously see water wants to go to the side of the membrane where the sugar is concentrated and dilute that out. So you probably don't think of osmosis as life-sustaining, do you? It is. As future nurses, I want to ask you a question. When someone's dehydrated, and say they're unconscious, okay? Maybe they lost blood, maybe they're just severely dehydrated, but they don't have enough fluid in their body. So you give them an IV fluid bag of sterile isotonic saline, okay? Or physiological saline. Sometimes that's called normal saline. Why can't you just give them an IV bag of sterile water to hydrate them? You know why? Because osmosis will kill them if you do that, all right? Osmosis would kill the person's red blood cells and all the other cells that they have in their bloodstream because of osmosis. So let me explain why osmosis is life-sustaining, and it's important that when someone's dehydrated, you give them sterile, physiological, or isotonic saline. It has all to do with osmosis in cells. So let's move on to the next topic. Osmosis and where water moves across the cell membrane depends on the tonicity or concentration of solutes that that cell is surrounded by. And that will dictate where water goes because there has to be a solute concentration gradient. So let's look at the first scenario. A hypertonic solution means a cell is sitting in a solute concentration that's higher than what's inside the cell. So what happens is water wants to go and dilute solutes. So if there's more solutes outside the cell, the water in the cell will leave the cell and go out into the environment to dilute those solutes. What will happen is the cell will shrivel up like a raisin and the cell will be dead. Okay? All right. The next tonicity is going to be a hypotonic solution. Now, in this case, you put a cell in a solute concentration that's more dilute than what's inside the cell. An analogy would be putting a red blood cell in pure, sterile water, which is what would happen if you gave someone an IV bag of sterile water. You're exposing all the red blood cells and white blood cells and everything else to a hypotonic solution. What's going to happen is water around the cell is going to enter the cell, which is more concentrated with solutes, and it's going to swell that cell up and cause it to burst. And that also causes cell death. That is why you don't do an IV bag of pure water. You would kill someone within 30 minutes, probably less. So what you want to give someone is an isotonic saline solution, and it's 0.9% saline. Oops, let me go back, get my laser pointer here. 0.9% saline. That is the same saline concentration that's already in your plasma. So what's happening is now someone's red blood cells are surrounded by fluid that has the same solute concentration as is inside the cell. And when the concentration on either side of a membrane is exactly the same, water doesn't go anywhere. It doesn't leave the cell, it doesn't enter the cell, and the cell maintains the same shape and size and continues to function. So isotonic saline is given to patients to hydrate them because of osmosis and tonicity of the solution. All right, so osmosis is pretty important. So let's look at a little GIF about that. So what we have on the left here is we have a cell that is in a hypotonic solution. Imagine that cell is in pure water. The cell contents are more concentrated with salts and proteins and sugars, so water in the environment fills the cell up and the cell goes and can lyse and explode. Okay, now the opposite extreme over here is if you put a cell in a very salty or sugary solution. What's going to happen is the water inside the cell leaves and the cell shrivels up like a raisin and is going to die. And the happy red blood cell here is in an isotonic solution where the concentration of whatever solutes are exactly the same and water doesn't go anywhere and the cell maintains its shape and form. All right, so let's move on to the last form of passive transport. Filtration. Filtration is pretty important. And before now, I've been talking about things moving across the cell membrane based on solute concentration on either side of a membrane, moving from high to low concentration. And even with water, it will go to the side of a membrane that has more solutes dissolved in it. So, but that is based on a solute and water concentration moving with a concentration gradient. Filtration is not based on solute concentration, it's based on blood pressure. So in your kidneys, what you've got is millions and millions of nephrons. And the start of a nephron is this thing called the Bowman's capsule with this filtering glomerulus right here where arterial blood enters your nephron. And the Bowman's capsule membrane is semi-permeable. It will allow small things to go through, like water, like gases, like glucose and small proteins, but big things can't fit through the pores of that membrane. Now what happens is arterial blood comes into the glomerulus under blood pressure, and that pressure forces the small things through the membrane into the filtrate. So up here, when substances move across the membrane due to fluid pressure being higher on one side of the membrane than the other, inside the Bowman's capsule, fluid pressure is very low. Outside in the arterial vessel called the afferent arteriole, blood pressure is high, and that is what is moving substances across the membrane. So filtration, think of this as your kidneys filtering your blood based on high pressure in your arteries and low pressure in your nephrons. Okay, so this is a good place to stop for Monday, okay? So and to recap, we talked about how permeable a membrane is. I'm gonna stop, and this will give me a chance to go back to your textbook. When I have, like I'm showing here, a page number in your textbook, let me give you an example. If I was continuing on to active transport, which I'm not today, what I would do is I'd say, oh, I can read the corresponding material in the textbook. I can type in page 17. So I'm going to escape out of this PowerPoint and I'm going to go to my textbook and I'm going to type in page 17 and lo and behold, active transport. Look at that, okay? So when I have that, please use those page numbers as a speed dial, so to speak, to go exactly to that place in the textbook so you don't have to surf through everything, okay? So, okay, that's plenty for today. Recap. Membranes are either freely permeable to things like gases. Most of our membranes are selectively semi-permeable that will allow small things through, but they will be more discerning with other things like large particles or charged ions, things even like glucose need transport carriers. Then we talked about the different types of passive transport. Simple diffusion, how gases can just move right across the membrane. We talked about facilitated diffusion as well, okay? We talked about osmosis, okay? We talked about transport with carriers. We talked about filtration. All of this is passive transport. You're going from areas of high concentration to a side of the membrane where it's in low concentration. Except in filtration, you're moving from high blood pressure to low blood pressure. Now, on Wednesday, we'll pick up with active transport, and that won't be a very long lecture. Okay, so that's plenty for today. I will talk to you later, and I've recorded this, so we are done for today. All right, happy Monday to you.